Welcome to this lecture and welcome to Let's Crack NEET UG English. I'm Neela Bakore, your biology teacher, and our topic which we are continuing with is the same, cell division, and we will start with meiosis I. Though we have talked about the first two stages of prophase I of meiosis I in the previous lecture, but that was just a brief introduction. We are going to be talking about those stages in detail again, so that in case if you have not understood the basic, you will be able to do that. But before we start, let us check the basic things. The audio, is it clear to everyone? Quickly start messaging yes or no, so that we can get started with our lecture. This topic is very important. There are many stages and there are many questions which are asked from meiosis I, and that too specifically prophase I. Okay, so this is what it is. Let me check. Good morning. Hi, okay, it is clear. Okay, so audio is clear. So we can get started now. All right. Okay, so meiosis I is what we are starting with, the prophase I. What we have done in the previous lecture is I gave you a complete overview of meiosis. And just a quick recap, meiosis is completed in two parts, meiosis I and II. In both meiosis I and II, there are two stages, that is karyokinesis followed by cytokinesis. But to make our life simple and to remember which stage is what, we have numbered it. So every stage which is in meiosis I has 1 in front of it, and it is Roman 1, and when the terms are used for meiosis II, we write 2, like prophase I, prophase II, and so on and so forth. So it is less confusing for us. Before we get started, we are on Let's Crack NEET UG English, but I'm giving you a heads up that I'm going to be starting my lectures on more channels. Like I have already started on Let's Crack NEET UG. I'll be starting on Biology 101, and I will be starting the special classes very soon. And on all these different channels, I'm going to start different topics. So what I'm going to do is, in the next lecture, probably I'll give you a complete overview of all the classes, the chapters, and the timings, so that whichever topic you think is the one which you need to study, you can start with that. And don't forget to install the Unacademy learning app, because this is where we will be getting all those things which I have just talked of. That is, the daily live classes will be available on various channels. You will have unlimited access to all those lectures, not only my lectures, but the lectures which are prepared by the other educators also. Like there are many top educators, so you can get access to all those. And this point is one which I'm talking about on a daily basis, like in every lecture, that plan your subscription wisely so that you are able to learn the complete thing in a proper manner. And it is always advisable to go for a long-term plan, because apart from the financial advantage, you also get the advantage of learning from the same educator, and you are able to develop that frequency, and you know, it really works well. And then there are the tests, and assessment is easier. So depending upon which grade or class you are in, you can go for a 12-month or a 24-month plan. And the most important thing is this code. So if you want to avail a special discount, you need to use this code of mine. All right. Okay, so we are on our topic now, that is Meiosis I, Prophase I. As I told you earlier that in cell division part, we do not label things. Everything should be self-explanatory. And how is it self-explanatory? It is possible only when you make a diagram which tells you everything. So you don't have to label anything. And there are stages where we have to give all the changes. So overview we have discussed Meiosis I, Meiosis II. In Meiosis I, we will have Karyokinesis I, Cytokinesis I. In Karyokinesis I, we'll have Prophase I, Metaphase I, Anaphase I, Telophase I. In Prophase I, we are talking of 5 sub-phases. So this is where we are starting from. So the first phase of Prophase I is Leptotene, and we have talked about this stage in brief in the previous lecture, but we will go over it. I'm going to start with a cell, and we will take animal cell, and then towards the end again we will differentiate between meiosis in animal cell and plant cell. So this is our cell, animal cell, which has undergone interphase. And if a cell has undergone interphase, that means G1, S and G2 phases are complete. And when we say all these phases are complete, that means every cell has double DNA. So here we are starting with a diploid cell, and again to make our diagrams easy to understand, we will start with the chromosome number 4. And here every chromosome is having sister chromatid, that we have to remember. Nucleus is large because it has already undergone interphase. It is an animal cell, so this animal cell has centriole, because this we need to draw and understand how these the centriole and the nuclear membrane and all those things are changing. Let us make the chromatin fibers here. So 1, 2, 3 and 4. This is the 4 chromosome that we are talking about. Now instead of chromosome, they are visible in the form of fibers. All the processes, changes will start now. Now this interphase cell enters Prophase I, and in Prophase I it enters the first sub-phase, that is our Leptotene. So let us make this cell again, which is in Leptotene. The nuclear membrane is still intact as it is. No dissociation of nuclear membrane, no dissociation of membranes of the organelle, nothing is going to happen in this stage. Centriole is also as it is, the splitting has already taken place in S phase, but here it is just the normal centriole or centrosome. Chromatin fibers, they have started to condense, and the condensation is not uniform. They condense at intervals, and all the chromatin fibers, they remain attached to a small part of nuclear membrane from their one end. So one end is attached here, the other end is free, and the thickening or the condensation is taking place at intervals. Now because of this, our fibers have beaded appearance. It appears as if there is a thread and there are beads. Which stage are we talking about? Leptotene, and we don't have to put the number 1 or 2 because this term or this subphase is only once. So during Leptotene, what all changes have taken place? Any change in the nuclear membrane, any change in the centrosome or centriole position? The answer is no. Nucleus or nuclear membrane is as it is, centriole is at its own position, organelle, which though we are not drawing, but they are also intact. So what has happened? here condensation of chromatin fibers has begun. But we have to specify that condensation is at intervals, and because of which, so the fibers have beaded appearance. If you remember, I told you one more thing about this stage in our previous lecture, that was that all these fibers are attached to the nuclear membrane, a small part of nuclear membrane by their one end, and the other end is free. So if one end is attached, that means all the fibers are focused at or concentrated at one point, and the upper ends are free. So it gives an appearance of bouquet stage. So all fibers are attached to a small part of nuclear membrane and free from the other end. And so the stage or these fibers, they look like bouquet. So the stage is known as bouquet stage. So leptotene is also known as bouquet stage. Why this name leptotene? Because leptos means thin, teen or nema means thread. So this stage is also known as leptonema stage, leptotene stage or bouquet stage because of the appearance of these chromatin fibers. Why leptotene or leptonema? Leptos or lepto means thin, leptos actually, so thin, and nema is for thread. So it is a thin thread stage. Because of the arrangement of the fibers, it appears like a bouquet. So it is a bouquet stage. Okay. So these are the changes which have taken place in leptotene. Nuclear membrane, organellar membrane, nucleolus, what all structures are there, they are still intact. We are not drawing all those structures here because our main focus is only on the chromosomes and the nuclear division. So the first subphase of prophase I. Is it clear? Okay. Let us take the next stage, zygotene. Zygotene is also known as zygonema. And zygo word comes from the pairing, because when we talk of zygote, it is formed by the fusion of the male and the female gametes. So in case of zygotene or zygonema, we are talking about pairing of threads or chromosomes. So again, what we will do is, we will draw the cell and we will see what all changes take place in zygotene. Nuclear membrane still intact, very prominently visible, no change, no dissociation. Centriole, centrosome is at the same place, though it has undergone splitting in S phase, but the two split centrioles are still at the same place. They have not moved to the poles. The chromatin fibers are going to continue their condensation process, and as condensation takes place, now you will start seeing the chromosomes. But here, because it is zygotene stage, we will have to show the pairing. So we started with, let us choose the color which we were using for our chromosomes. So this is our one pair, and we will draw two because we started with 4 chromosomes. So if there are total 4, there are actually 2 pairs. So we have drawn these 2 pairs, and now what we are going to do is we'll draw these 2 pairs outside also in detail, and then we will write down things. So this is our one pair of chromosomes. Such a pair is called homologous pair. Can we call any pair of chromosome as homologous pair? There has to be something specific for homologous. Okay. Wait, let us wait. The doubt. Ayush Soni has written a doubt thing. Where is the doubt? Amulya is saying that I should be talking in Hindi. I think you've joined a little late. Let me tell you that I'm taking lectures on Let's Crack NEET UG, which is a bilingual one, Hindi-English mix. Then this channel, which is pure English, and then I will be starting my lectures on Biology 101, which is again going to be Hindi-English mix, and the special classes also. So we are talking about all types of students. Those who are comfortable in pure English, they can watch here. And in the beginning I told you that the language or English which I use to explain the concepts of biology is very simple. So even if it is English, the terminology anyways, whether you study in Hindi-English mix, remains the same. Okay. Disintegration happens where? This is what is Ayush asking me. Disintegration of what? Nuclear membrane has not dissociated so far. We will talk about when it dissociates. We are just starting the Prophase I. We have talked about the changes which take place in Leptotene and Zygotene. Let us reach the end part and we will see where this dissociation takes place. Okay, so let us first finish all the stages, and then we will talk of this dissociation part. Okay, so I was talking about this pairing. So this pair is called homologous pair. Homologous pair is a pair which has the same genes at the same locus. Now this is little specific terminology which I am using, but it is important because when we start genetics, at that time also we will be using these terms. So I'll give you a little description of what we mean by this homologous pair and same genes and same locus separately in one corner so that it doesn't interfere with our stage. So if suppose this is one pair and we know that for every trait or every character there is a gene. There is a gene for height, there is a gene for eye color, there is a gene for shape of the nose, for everything. If you're talking about the plant, there is a gene for height of the plant, there is a gene for the flower color, and so on. So suppose this chromosome has a gene for height, and this chromosome has been received from the male gamete. The chromosome which is received from the female gamete will have the gene for height at the same position. So position becomes the locus, and now one gene for one character can have two forms which are known as the alleles. The alleles can be dominant or recessive. So suppose here we write that it has dominant allele, and at the same locus of the other chromosome it is a recessive allele. So gene is same, it is the gene for height, but from one gamete the allele for this gene which is received is the dominant allele, and from the other the one which is received is the recessive allele. Both can be dominant, both can be recessive, one can be dominant and other can be recessive. But such a pair of chromosomes which has the same genes at the same position, then that pair is known as homologous pair. And the two chromosomes which are there in the homologous pair, one out of these two has been received from the male gamete and the other has been received from the female gamete. So in sexual reproduction, when fertilization takes place, sperm or the male gamete brings one such chromosome and the egg or the female gamete brings the other same type of chromosome. This is homologous, and this pairing has taken place. So here we will write that what is the change or zygotene in zygotene stage? Pairing of homologous chromosome takes place. What is this pairing known as? This pairing is also termed as synapses. So there is synapses or synapse formation. So when we say synapses formed, it means pairing has taken place. Now these two chromosomes, they are together. We have to remember that every chromosome has double DNA, or in other words, every chromosome has two sister chromatids. So now what is going to be visible to us? Let us make this diagram a little bigger and in more detail. This is the chromatid, and here we are seeing these two sister chromatids. Here is a chromosome and we see those sister chromatids. Same is going to be in the other chromosome, and we are not drawing the DNA which is inside because that part we have already understood. Okay, so how many chromosomes are visible to us? Only two. So this is a pair. One more stage. Now these sister chromatids are going to separate. So the chromosome is like this, it is going to separate. So after separation, it will be visible to us like this. These two sister chromatids have separated. We will go over this one more time because this is the most important concept that we need to know. In leptotene, condensation begins. That means the chromatin fibers, they start to become thicker as condensation has started. So in zygotene, now you are starting to see the little rod-like chromosomes which are visible here. Every chromosome has two sister chromatids. Remember what happened in S phase of interphase? DNA replication. So the pairing takes place first. What is this pairing of? It is a pairing of homologous chromosomes. What are homologous chromosomes? Homologous pair is a pair of chromosomes which have the same genes at the same locus. We have understood this concept, what we mean by same gene and same locus. Chromosomes pair, and this is called synapses. Why are we trying, or why is the cell trying to do all these things? Because in prophase there will be exchange of genetic material. Now if the exchange of genetic material between these two chromosomes has to take place, there should be crossing over. So suppose we talk about that these are two chromosomes and crossing over has to take place. So first the chromatids will split into two arms. You will call these two as sister chromatids. Let us label this first. These two are sister chromatids and these two are also sister chromatids. Okay, so now what has happened is these two chromosomes, they come closer, we see a pair. Now chromatid of one, that is the arm of one chromosome splits, the arm of the other chromosome splits. These two are called sister chromatids and these two are also called sister chromatids. Now when crossing over takes place, one sister chromatid here and one, they cross. So if they cross, then there will be exchange of genetic material. But for this, the two chromosomes should remain together. If the two chromosomes are apart, how will crossing over take place? So what is that structure which is going to keep these two chromosomes together? That structure is called synaptonemal complex. So between these chromosomes, there is a network of proteins which is formed, and this network of proteins is known as synaptonemal complex. What is the significance of this? The purpose of synaptonemal complex is to keep the two chromosomes of the homologous pair together so that crossing over can take place. So what is our next change? The next change is synaptonemal complex is formed. And this synaptonemal complex is going to hold these two chromosomes together. In this case, once the sister chromatids are visible, on every side we see four arms. Two sister chromatids of one chromosome and two sister chromatids of the other. This is called a tetrad. The pair was called bivalent. Now we can add one more stage here. What can we call this? Only this part. This is known as a dyad. You know, this is very, very important. Once we finish this part, we will have some questions. Suppose there are, let us take this example so that when we have a complicated question, it will be easier for us. We are starting with one homologous pair. If there is one homologous pair, how many tetrads are going to be visible? Only one. So this complete stage where every chromosome is showing two sister chromatids is called a tetrad. So in one homologous pair, you will see only one tetrad. But in one homologous pair, which is now seen in the form of a tetrad, how many dyads do you have? You have two dyads. So such type of questions can be asked. So how many bivalents? One. How many tetrad? One. But how many dyads? Two. So if we increase the number of homologous pairs, we will be able to answer this question. So now, recap. Homologous chromosomes pairing takes place, which is called synapsis. To hold these two chromosomes together, there is a synaptonemal complex which is formed. The chromatids split and you start seeing sister chromatids. What can we call these two? Suppose I am talking about this one and this arm. What should these two be called? They are called non-sister chromatids. The arms of one chromosome, they will be called sister chromatids, and now we are talking about one arm of one chromosome and the one arm of other chromosome. This becomes non-sister chromatids. So what is the next change which is visible in this stage? Sister chromatids appear, and so dyads and tetrad are visible. All these changes are taking place inside the nucleus. So I have shown you the changes which take place inside the nucleus, but we have drawn it outside. So what we will do is, in this case, we will make one more arm here, so that our tetrad is visible, and we will keep one homologous pair as it is, so that this transition is clear. So one pair is shown as homologous pair, and then after all the splitting and everything we are showing the tetrad. So here we are showing the bivalent, and here after the changes we are showing the tetrad. Okay, but how exactly it happens, that we have drawn separately. Let us not forget that our synaptonemal complex is still existing. As long as you want these chromosomes to remain together, you have to have that synaptonemal complex. But moving forward, we may not draw it because we are going to show some other changes. But we have to remember till we write that synaptonemal complex dissociates. So here, pairing of homologous chromosome has taken place. The pair is called a synapse or a bivalent. Then the chromatids split. You start seeing sister chromatids. One chromosome with two sister chromatids on each side you would call dyad. And when pair splits, you see 4 arms on each side. That is what we call the tetrad. So these are the changes which are visible in zygotene. Second stage done. Nuclear membrane is as it is, nucleolus, organelle, everything same, intact. Centriole, though undergone splitting in S phase, but still at one pole. Changes clear? Okay, it says explain it once more. What we'll do is, let us do one thing. Let us finish the 5 subphases of Prophase I, and then we will have a quick recap of everything, so that in case if you are missed out on anything, that part will be clear. So now let us move to the next stage, that is Pachytene. The basic structure we will draw first. This is our cell, which is now entering Pachytene. The nuclear membrane is as it is. No dissociation of nuclear membrane. No dissociation of organellar membrane. Centriole, centrosome at its position, not changed. What is inside? In the center or inside the nucleus, we have those chromosomes. So this is a chromosome with its sister chromatid. This is another chromosome with its sister chromatids. Okay, we will draw it outside also. This is the chromosome with two arms and 2 arms. This is the other chromosome with its 2 arms. Okay, to differentiate these 2 chromosomes, I am going to make one chromosome solid and other chromosome hollow, just for our understanding, because we want to see how this crossing over takes place. Are these 2 chromosomes easily distinguishable? One is empty, hollow, and the other is solid, so that when we show the exchanged genetic material, it will be easier for us. What happens in one pair, similar things are going to happen in the other pair. Now, let us try to visualize what is happening. In zygotene, the 2 chromosomes paired, right? That was homologous chromosome, synapses. Now these 2 chromosomes are held together by the synaptonemal complex. The 2 chromatids have split and you start seeing the sister chromatids. Now in Pachytene, the two non-sister chromatids which have to undergo exchange of genetic material or crossing over, they have to come closer, and you should start seeing some cross kind of a thing. Now from the front side you would see that there is an X-shaped body or a cross, but if I change the orientation, it is like they have not fused yet. So what is happening here is you are seeing a cross-like structure or X-like structure from the front side, but actually these two non-sister chromatids have not fused. If I change the orientation, you see a gap, but if they have to cross over, they will have to fuse here, which is going to happen in the next stage. But before they fuse, they should actually come closer and make that X-shaped structure appear to us. So this X-shaped structure later on, when it is completely formed, will be called chiasma or chiasmata. So here what we write is the two non-sister chromatids come closer and X-shaped structure starts to appear. This structure is called chiasma or chiasmata. So what we say is in this stage that chiasma formation starts. When is the structure called chiasma? Is when the crossing, when they fuse, that complete fused structure, X-like structure, that is called chiasma. But before these can fuse, they have to come closer, overlap each other, then only this chiasma will form. So the sister chromatids are like this. Non-sister chromatids move and you see an X-like body. But fusion has not taken place so far. So chiasma formation starts, and if you carefully observe this part, this sister chromatid has its own genetic material, and the blank one or the hollow one which we have shown has its own genetic material. Exchange has not taken place. So here we write chiasma formation starts. So that X-shaped structure starts to appear. So what we'll do is with the other chromosome, we will show this X-like structure, and here also I'm going to make one chromosome solid so that it can be easily distinguished. Okay. At how many places can crossing over take place? Crossing over can take place maximum at 4 places between the homologous pair. So if this is the homologous pair, the splitting has taken place. So crossing over can be between the two non-sister chromatids and these two non-sister chromatids, and this is on the upper end. When we are talking about the lower side, again the crossing over will take place. So there are 4 possible places where crossing over can take place, but the most common number is 2. But maximum, theoretically if we are talking about, it can be at 4 places. But normally what is most commonly seen is at 1 or 2 places where crossing over takes place. And now comes the next stage in which the maximum changes are going to take place. One thing which we normally don't draw here, but we have to remember, and that is the protein, that is synaptonemal complex, which are holding these 2 chromosomes together. What was the significance of these fibers, the synaptonemal complex? So that the chromosomes are held together and the non-sister chromatids are able to cross over. If these fibers are not formed, the chromosome, because the chromosomes can move anywhere in the nucleus, so they can be apart, and if they are apart, crossing over will not take place. So is this stage clear now? Let us come to the next stage, Diplotene. Again, we will draw our basic cell and the structure which is there. This is our cell in Diplotene, and the nuclear membrane is still intact. No dissociation of nuclear membrane. What is the position of our centriole or centrosome? It is still at one position. And now let us talk about our chromosomes. So our chromosomes, I'm going to draw the chromosome here. This is one chromosome showing its sister chromatids, and this is the other chromosome which we are showing solid. We will make it solid, okay? And now try to find out or identify the difference which is between this stage and your previous stage. I'm going to show you the previous slide also. Concentrate on the cross. That means this is the place we are talking about. Okay, have you seen this one? Let us go back to the previous. Is there any difference between the two cross-like structures which we have shown in Pachytene and now in Diplotene? I'm going to draw it here for you again. In Pachytene, what we showed was a cross. One chromosome was hollow, other chromosome was solid. This was the cross-like. And how is it different in this stage? Same two chromosomes. Let us make this one solid. Okay, is the difference clear now between this stage? This is in the previous one, that is Zygotene. Sorry, this is in Pachytene. In Pachytene, and this one is Diplotene. Is there any difference? In this cross, this is the point which we have to observe. In Pachytene, the hollow chromosome is completely having its genetic material in the the arm we are talking about, non-sister chromatids, and this chromatid is having its own genetic material. But here in the next stage, the hollow one arm has a little part of the solid chromosome and the solid arm has a little part of the hollow chromosome. So has the exchange of genetic material taken place? Then only the chromosome, that piece is going to get exchanged. So this structure where crossing over takes place. So now what has happened is, if we have to recap, in Zygotene pairing took place, non-sister chromatids, sister chromatids are visible. Now in Pachytene, you start seeing this cross-like structure, but the fusion has not taken place. In Diplotene, these non-sister chromatids actually fuse here, and this point is called recombination nodule or chiasma or chiasmata. The synaptonemal complex is still there, and this was the purpose for which those synaptonemal complexes or those threads were formed, so that this exchange of genetic material takes place. So what are the changes here? Here we say recombination nodule is formed. An exchange of genetic material takes place. Exchange of genetic material takes place. And this exchange is actually an enzyme-driven process. So the enzyme which helps in this exchange is called recombinase. Recombination, that's why recombinase. So enzyme recombinase helps in exchange of genetic material. And this complete process of exchange of genetic material with the help of this is known as crossing over. So here we say crossing over is completed, recombination is completed, chiasma formation is completed, everything is completed here. Synaptonemal complex is still there. So now exchange has taken place. Exchange takes place always between non-sister chromatids. This we have to remember, very important. Crossing over takes place between non-sister chromatids. Always. It can never take place between sister chromatids, because sister chromatids have the same genetic material. It was the original genetic material which was copied in S phase. So there is no significance if the same copies get exchanged. So it is always between the non-sister chromatids. So exchange of genetic material has taken place. So now if we make this chromosome inside the nucleus, we started with 2 pairs, so we have to show those 2 pairs here with exchange genetic material, but we have understood the process in detail, what exactly is happening in this process of exchange of genetic material. This is the solid part and there is an exchange. So this exchange should be visible between the Pachytene stage and the Diplotene stage. Okay, in Pachytene stage the chiasma formation just starts, so you are able to see that cross-like structure, but exchange has not taken place because the 2 sister chromatids have not fused yet. This enzyme-driven process of recombination or crossing over takes place in Diplotene. Here, the main events of Prophase I are completed. Now comes the last phase, subphase of Prophase I. So what happens in the last phase? That is diakinesis. Everything that happens in prophase of mitotic division, everything is going to happen here. Now what we see, we can have an early stage and we can have a late stage. But what happens in prophase, I'm sure you have revised that mitotic division that we talked of. In prophase, that is only of mitosis that we are talking about. Nuclear membrane dissociates. Nucleolus or membranes of the cell organelle, they all dissociate. Chromatin fibers condense. Aster rays appear, they assemble and they become visible. Spindle fibers assemble and appear. All those things will take place in diakinesis. So everything what happens in prophase of mitosis, same thing happens here. Now, a question might arise that chromosomes are already condensed, and in prophase we say chromatin fibers condense, right? Here, by the end of Diplotene, When this crossing over takes place, there are two more changes which are taking place. Up till now, how are the chromosomes? If we are talking about just the Diplotene stage, exchange of genetic material has taken place. These two non-sister chromatids are fused here. Recombination nodule is formed. That means exchange of genetic material has taken place. Do you still need the synaptonemal complex? The purpose was that the chromosomes are held together. Sorry, they are held together and this chiasma can form. Now chiasma is formed. The two non-sister chromatids are fused. So now the synaptonemal complex starts to dissociate. When synaptonemal complex dissociates, these two chromosomes which were held together, they start moving apart. Okay, so in this part we will add one point, which the process which actually starts here, that is synaptonemal complex start to dissociate. It starts because their job is done. And for a very short period of time, the chromosomes will decondense. They become thinner. And now why do we want to see those two sister chromatids separately? So they are again fused. So this also begins here, that is decondensation and the joining of the two sister chromatids, except for this point. So this point will remain, and here, like if I have to show what exactly is happening, is this chromosome will appear like this and this chromosome will appear like this. Okay, so these two are fused and this is visible like this. These two are fused and it is visible like this. But here chiasma formation is there, exchange of genetic material has taken place, so that chiasma is going to be visible. So sister chromatids start to disappear, they start to fuse. Synaptonemal complex will start to dissociate because their job is done, but you still see chiasma. How was chiasma formed? The non-sister chromatids, they overlapped and then they fused. Exchange has taken place. Now how is this cross or chiasma going to separate? How will these two chromosomes separate? The two chromosomes are not going to separate like this, the way they fused. They are going to separate like they move from this point, slowly they come to the end and then they separate. This process is called terminalization. The process starts in Diplotene and gets completed in Diakinesis. Okay, so let us come to the stage of ours, Diakinesis, and let us see what all changes are taking place here. We will draw our cell first. What is going to happen to the nuclear membrane? We don't have to show early stage and late stage because we know what is happening. It's a slow process. So nuclear membrane dissociates, nucleolus dissociates, all organelle membrane dissociate. So you would not see the nuclear membrane here. What is going to happen to the centriole? The centriole has already undergone splitting in S phase. One remains at one pole and other would reach at the other pole. Okay? Aster rays and spindle fibers would appear. Okay, we'll have to use a different color for our spindle fibers. The smaller rays are the aster rays, and then we will see the spindle fibers. This is how the spindle fibers are going to be. the same type of spindle fibers, chromosomal and the continuous one. So this one is the continuous spindle fiber. The smaller rays are the aster rays. All these things we already know, and this structure, the blue structure which we have drawn, is the centriole, and together this structure is known as aster. So here is one aster and the other aster is at the other pole. This is animal cell, so we are seeing two asters. Well, our focus is not this right now. Let us come to our chromosomes. Okay, so our chromosomes, this is one chromosome. Synaptonemal complex has dissociated. Terminalization has been completed here. I'm going to show you the stages of terminalization. Suppose this is the chromosome where we are seeing the chiasma. This is the solid chromosome, and this much part has been exchanged. When terminalization takes place, the next stage is going to be everything same. And the last stage is this is wait. Okay, now is it clear? The three stages. Here in the first stage, you are seeing a big X-like structure, the cross, this much big. In the next part, it has come to very close to the end, and here it is already separated with exchanged genetic material. So as I said, terminalization starts from the point where crossing over takes place, and then it moves closer to the tip. Slowly these two arms will get separated. So this is known as terminalization. So here we say that terminalization is completed. And how do they separate? It is in a zipper manner. In our jackets, there are zip, chain. Now every chain has three parts. There are two chains, two halves rather of that chain. One is on one flap and the other is on the other flap, and there is a clamp. Suppose these are the two halves and you want to separate these two halves and the clamp is in the middle. Can you remove the clamp from the middle? Not possible. So you have to move the clamp till the end, and then only these two flaps separate. So this is how the two non-sister chromatids are going to separate. So how do they separate? Terminalization takes place by zipper manner. Now this zipper manner is for our understanding. It's not a technical term, but how exactly it's going to take place, because chiasma formation took place by crossing like this. The two non-sister chromatids come like this and they fuse. Separation doesn't take place like this. Separation takes place when the two arms come to the end and then they separate. So terminalization is going to get completed here. Let us see what is the position of our chromosome. This chromosome has the exchanged genetic material. This is another chromosome. This is that solid one which is having the part of the hollow chromosome to attach to it. This is one more chromosome. It also has that exchanged part, and here is one more chromosome which is having this exchanged part. You still see 4 chromosomes, and these 4 chromosomes are with exchange sorry. Okay, with exchanged genetic material. So what all things are happening here? End of prophase I. Nuclear membrane, nucleolus, membrane of organelle, dissociate, esterase, spindle fibers assemble and are visible, clearly visible. Chromosomes are with their exchanged genetic material. Prophase I is now complete. So we say prophase I is the longest phase of meiosis. And why do we say so? Because there are so many things which are happening here. Okay, and as I told you, once we complete this, we are going to have a recap of this. So let's go back to our first slide. Vaibhav is saying, ma'am, reply karo padte some questions. You know, the basic thing is, if you understand or listen to the complete process, invariably the doubts automatically get cleared. And that's why I told you that let me finish prophase I and then we will come back to it, so that I'm sure 80% of your doubts are over now. But still, in case if you have something, we will go over this. Okay, we are starting with prophase I. First is leptotene, which is called the thin thread stage, or leptonema, or bouquet stage. Here, the nuclear membrane does not dissociate. Everything what happens in prophase of mitosis happens towards the end of prophase I of meiosis I. But chromatin fibers are going to condense, so here we see condensation, and condensation is not uniform, so it is at some places. So the thread, it gets condensed at one place, then is thin, then again thick, so it gives a beaded appearance to the chromatin fibers. These chromatin fibers, they remain attached to the nuclear membrane, a small part of nuclear membrane, and the other end of these fibers is free. So it gives a bouquet-like appearance. So now we know the reason for all the names or terms which are given to this stage. Leptos is thin, nema is thread, bouquet stage because of the appearance. Okay, so far this stage clear? We have started with a cell, an animal cell, which has completed interphase, that is G1, S and G2, and the chromosome number that we have taken 2n as 4. The next stage, zygotene. Total chromosomes were 4, and these 4 can be written as 2 pairs. Such a pair which has same genes at the same locus is known as homologous pair. So in zygotene, there is going to be pairing of homologous chromosomes. You want these homologous chromosomes together because later on exchange has to take place. Exchange takes place between non-sister chromatids, so these 2 homologous chromosomes should be together. When these chromosomes are together, something should hold them together, otherwise they will move in the nucleoplasm here and there. So to keep them together, synaptonemal complex is formed. Now, each chromatid of a chromosome, which is like this, splits. If you remember in the chromatids, there was an original DNA and a copied DNA. So when these split, this arm gets original DNA, this gets copied DNA. You call these two sister chromatids. So when there are two chromosomes, you would see two sister chromatids of this, two sister of this. So at one end there will be 4 arms visible, 4 chromatids, and on the other end also 4 chromatids will be visible. So when you see 4 arms at one end, then you start calling it a tetrad. So one homologous pair is known as a bivalent. One chromosome, when splits into its sister chromatid, that is known as a dyad. And when there are 2 dyads, because it is a pair, then that is known as a tetrad. So in zygotene, synaptonemal complex is formed, sister chromatids are visible. These are the important changes. No change in the nuclear membrane or anything. Pachytene. Here, the process of crossing over actually starts. How is the process going to start? These non-sister chromatids have to cross. Exchange of genetic material has to take place. For that, they should make a cross-like structure. So they start to move, and from a distance X-like body is seen. But actually there is no fusion. So we say the process begins. What is the process? Crossing over process, exchange process. It begins. Recombination nodule is not formed yet. So this is a shorter stage. Everything major takes place in Diplotene. Now the cross which was visible, these 2 non-sister chromatids actually fuse. That is called recombination nodule. So recombination nodule is formed. With the help of an enzyme, recombinase. And when this recombination nodule is formed, exchange of genetic material takes place, and this complete process is known as crossing over. So this takes place here. When these 2 are fused, we do not need the synaptonemal complex now. So synaptonemal complex starts to dissociate. Its job is over. The non-sister chromatids which are not crossing over, they can fuse. So here we have shown this, that these 2 which were separate, now they have fused. So all these changes, they begin towards the end of Diplotene. Now comes the last stage, diakinesis. Everything that happens in prophase of mitosis happens here. Nuclear membrane, nucleolus, membrane of the organelle dissociate. Spindle fibers are formed, chromosomal and interpolar. And aster rays also form. So if aster rays are formed, you start seeing the 2 asters, and prophase is complete. But the crossing over is also over. So how do these non-sister chromatids which have formed a chiasma, they separate. They start moving towards the end, and then from the terminal end they get separated. This process is called terminalization. So this terminalization process is completed here, but it begins in the previous stage. So there is a gradual movement towards the tip. This is prophase I complete. So initially there were few questions. I hope all those questions have been now answered. Now metaphase I, that is of Meiosis I. So let us make our cell now. Our cell, the chromosomes they are going to come on the equatorial plane. But do you see a change? This is the chromosome with exchange genetic material. This is the chromosome with exchange genetic material. This is the chromosome with exchange genetic material, and this is the chromosome. Let us make the centriole here and centriole here. Let us also make the spindle fibers, the aster rays and the spindle fibers. In metaphase, chromosomes are going to come and align on the metaphase, but here chromosome pairs, that is homologous pairs, align on equatorial plane. Very, very important. And once chromosomal spindle fiber from one pole is attached to only one chromosome. So one chromosome of the pair is attached to only one chromosomal fiber from one pole. Okay? Now, I have explained Prophase I completely. I have given you the change which has taken place in Metaphase I also. I want you to complete next two stages. That is Anaphase I and Telophase I. Okay? So this is what I want to give you as your homework. You will complete this. You have the idea of complete mitosis. I have given you the first two stages of meiosis I, prophase in detail, metaphase also. And now just try to think what is going to happen in Anaphase I and Telophase I. This will help me also understand whether you have understood this complete process or not. And if you are able to do this, you can also write what happens in cytokinesis. Okay? So, I have given you 3 stages to be done on your own. If you've understood everything clearly, you should be able to solve those changes. And we will take up these 3 stages of your homework in the next lecture so that I know whether you've understood this thing clearly or not. And again, last thing, that whenever you subscribe and take any subscription plan, don't forget to use my code so that you get the discount, 10% discount. Revise all the stages, go over all the changes that we have discussed, and I'm sure you will be able to complete your homework before the next lecture. I'll see you in the next lecture. Till then, work on your homework. Bye-bye. Take care.